Hello and welcome to some biology class. Friends, today we shall discuss about ion channels. Ion channels are transmembrane proteins that facilitate ion flux across cellular membrane. They mediate the passive transport of a variety of ions down their electrochemical gradient. Ion channels mediate several essential biological processes such as cell volume regulation, swimming behavior in unicellular organisms such as paramecium, stomatal movement, leaf closing response in Mimosa plant, and also muscle contraction and neuron excitation. All ion channels share some of the common properties. They are ion selectivity, activation gating and inactivation gating. Ion selectivity means ion channels transport only specific ions, and in activation gating the opening of a channel is regulated, and in inactivation gating the closing of a channel is regulated. Ion channels are transmembrane proteins that are present in all eukaryotes from protists to fungi and also in plants and animals. The rate of transport mediated by ion channel is much faster. Ion channels can mediate the transport of more than 1 million ions per second. These transmembrane proteins mediate the transport of variety of cations and anions such as sodium, potassium, calcium, chloride and bicarbonate down their electrochemical gradient. So the transport facilitated by the ion channels are passive transport. Ion channels are divided into three families. One is voltage gated channel, second is ligand gated channel, and the third is mechanical gated channels. Voltage gated channels are activated by change in voltage across a membrane, whereas ligand gated channels are activated by various neurotransmitters such as N-acetylcholine, glutamic acid, serotonin and gamma aminobutyric acid. Structure of ion channels. Voltage gated sodium, potassium and calcium channels show a surprisingly high degree of similarity in their amino acid sequence, which suggests a common evolutionary origin. A four-fold symmetry has been suggested for these cation channels. The channels are composed of 4 subunits and each subunit is 600 amino acid long. Each subunit has 6 transmembrane helices and 2 antiparallel beta strands. The beta strands which line the pore are located between helices 5 and 6. This figure shows the structure of a single subunit of voltage gated potassium channels. These are the transmembrane helices. Totally 6 transmembrane helices are present, and the amino terminus and the carboxyl terminus are located on the cytoplasmic side, and between helices 5 and 6 there are 2 antiparallel beta strands lining the pore. Second type of ion channels are ligand gated channels. The channels are activated by neurotransmitters such as N-acetylcholine, glutamic acid and gamma aminobutyric acid. A five-fold symmetry has been suggested for these neurotransmitter activated channels. The channels are composed of 5 subunits, and the subunit contains specific binding site with which the neurotransmitter bind. This figure shows the structure of a acetylcholine receptor channel. In this channel you can see that there are 5 subunits, and the subunit contains specific binding site with which the acetylcholine can bind and activate the channel. The most important ion channel is the potassium channel present in the plasma membrane of animal cells. There are some potassium channels which remain open even in unstimulated cells. These potassium channels are called as potassium leaky channels. The channels play a critical role in maintaining the membrane potential. So friends, this is all about structure and function of different types of ion channels. For more update, please subscribe my YouTube channel, Sam Biology Class, and also please share your comments. Thank you very much.